Welcome to your first lecture on molecular genetics on the RNA and DNA. I'm Wendy Nofke and I'm going to be doing this presentation for you today. Some of your lectures will be presented by your co-instructor, Lauren Davis. One of the major milestones in molecular biology was coming up with what they call the central dogma of molecular biology, which is that we start out with our DNA. The DNA is used to make an RNA, and then the RNA is used to make a protein. Another person who was significant in helping us understand how the DNA works was Chargeoffs, and he found the concentrations of the bases adenine, thymine, guanine, and cytosine, which are abbreviated as A, T, G, and C, differed from one species to another. But within a species, he found that the concentrations of A and T were similar and the concentrations of G and C were similar. This is what's been known as Chargeoffs rules. This is significant because A pairs with T and G pairs with C. So you should have equal numbers of A and T and then equal numbers of G and C. DNA is in the shape of a double helix, and this was discovered by Watson and Crick. It consists of two polynucleotide chains. Each nucleotide is going to have a deoxyribose and a phosphate that are going to make up the backbone, or what we would think of as the sides of the ladder. Then we have nitrogen-containing bases, the A, T, G, and C, that are going to make up the rungs of the ladder. In this diagram here, you can see you've got your phosphate and your sugars that make up this backbone, and then your nitrogenous bases here making up the rings of the ladders. So A and G are what we call purines. They have a two-ring structure, and C and T are pyrimidines. They just have a single ring structure. So you're always going to have guanine and cytosine paired here, which are a purine and a pyrimidine paired. Also, you'll have A and T paired, and this results in having a purine and a pyrimidine as well. The process of DNA replication is what we call semiconservative. You're going to have two new strands formed, and each of those new strands is going to have half of the old strand. So we start out with our double-stranded DNA, and an enzyme is going to separate the bonds between the complementary bases. This is going to expose the inside of the bases. You'll have two new separate strands that are going to pair with new complementary bases. So you can see down here the new complementary bases are being added, and that will end up forming two new daughter strands. In the cells, especially eukaryotic cells, we're going to have both RNA and DNA. DNA is found in the nucleus, and it's needed to make the RNA. The RNA will go into the cytoplasm and be read by the ribosomes. RNA is single-stranded in eukaryotic cells, and its DNA is double-stranded. Here in this diagram, you've got RNA on the left and DNA on the right. RNA is going to use the nitrogenous base U, or uracil, instead of thymine. It's also going to contain the sugar ribose instead of deoxyribose. Our cells also have multiple types of RNA. The mRNA, or messenger RNA, is going to be created in the nucleus, and it will be sent out to the ribosomes in the cytoplasm. The rRNA is ribosomal RNA, and this helps form part of the structure of the ribosomes. The tRNA is your transfer RNA. This is going to help to bring the correcting amino acids to meet with the ribosomes in forming the proteins. So here we can see the mRNA being formed and being sent out to the cytoplasm, and here you have a ribosome on the cytoplasm reading that. Here you have a tRNA that's going to bring the amino acid in to form the polypeptide chain.